That's one thing we can do is have the removal of introns. What else can we do aside from remove some introns?
Are you talking about like elongation and stuff like that too?
No, that was just like the overall process. So remove introns. We could put the caps on, right? We put on the 5' cap, then the poly A tail, which of course is on the 3' end. Both of those are going to help to export or send out the mRNA. It also is going to assist in getting the process of reading the mRNA to begin. So that's good. So it helps export the mRNA and protect mRNA. There's a lot of enzymes that can break down RNA, but putting on those caps will help to facilitate that, or avoid that, better put. We talked about removing the introns and keeping the exons, right? That was the last picture we drew out yesterday. Remember that the introns are non-coding sequences. They don't code for amino acids specifically. Exons, of course, are coding regions. That will eventually be translated. And this process of removing the introns is called RNA splicing. And so RNA splicing is probably the next thing that I want to go into, which of course is the removal of introns. Remember, we remove the introns, we keep the exons. Cool. The unit that helps to facilitate this is called the spliceosome. If you look at this picture real quick, I had one, but then I thought, well, you probably don't want to draw it because it's Friday, like that Rebecca Black song. So basically you have these things called SNRPs, SNRNPs, and a couple of other items, a couple other proteins that come together and they become what's called the spliceosome. And that spliceosome is the unit that will help to remove. You'll see how kind of it does that removal of this intron section. It might be a little hard to see, but this is a pink section, whereas this is red in the exon section. So you're removing, yeah, basically the introns. Cool. Back up. One thing you'll get to, or we will get to actually in a little while, and we might as well mention it now, is there's something called alternative RNA splicing. Have y'all read about that? What is that called? Or what does that do? What does that mean? Okay. Well, let's talk about that. Alternative RNA splicing. And this is it's a hard concept, and it's one that scientists don't really quite understand why it's there. But in this type of splicing, introns can sometimes be exons. That seems a little awkward. So let's take a segment. Bless you. Of mRNA. Let's say that this is pre-mRNA, right? And let's put in some introns and exons. So my designation of E1, E2, and E3 represents that I have, or means that I have, 3 different exons, and these 2 are introns. Good. Now, that's though for one gene. You can understand that these 3 exons would come together and make our final sequence of mRNA, right? And we've learned that. We went over that yesterday. There's E1, there's E2, and there's E3. They're all together. But what I'm suggesting is that in some cases, we can put these in different orders. Sometimes introns could be exons, and you could create a totally different protein that might look something like this. You see what I've done here? Change the order up. This sequence of mRNA, though it's made of the same stuff, it's obviously not in the same order. It's theorized that in some cases this actually might be an I. So the thinking is that in some segments of mRNA that are translated into proteins, that sometimes it might go like this. Sometimes your introns aren't as worthless or as worth as much trash as they were in a separate protein or separate mRNA sequence. Does that make sense? This would be, to me, analogous if you think about maybe doing one of these 2 sequences. Remember, I view the production of mRNA as like making a note card from the library, because the DNA, of course, is the resources in the library. This would be nothing more than putting your note cards in different orders so that your paper is fairly the same, but maybe the order might mean just a little bit different to the teacher that's reading it. Right? So these 2 proteins would be very similar once they're produced, but they will be somewhat different. The other thing that's valuable a lot of times too is that when you have similar proteins, like with a similar function, they're usually coded together. So a lot of times we see, and this would go along with RNA splicing, proteins that work together are oftentimes coded in neighboring sequences or neighboring pieces of the DNA. A good example of that would be our lactose enzymes, or the lactose enzymes of a bacteria. To break down lactose, which we know is a simple 6-carbon sugar, or is it? It's a disaccharide, it's 12-carbon sugar, made of glucose and galactose, right? When we break that down, we need like 3 different enzymes to do it, not just one. So I want to make sure that you get that sometimes you would put the same ones together. Thank you. And let's look at this picture now. And I think this one you probably would want to just jot something down about, because this is where we're going next. You might want to use your different colors to go over the different exons and the different protein domains that are produced. Something very, very simple. And then we'll leave splicing.